My name's Ryan, and our host is Tomek. And as you know, the topic for today's webinar is, Tomek, can you go to the next slide? There we are. We are seeing your screen. That's good. Okay. Topic for today's webinar has to do with looking at steatosis and fibrosis in rodent models for liver disease. Tomek is our host. He's our CTO. But before we got into his introduction, I wanted to introduce the company, SonoVol, because I've seen the list of registrants, and I know some of you, but I don't know all of you. So I wanted to just formally introduce what we're doing here at SonoVol to you. So this is the team. We're a small team, but a dedicated group of hardware engineers, software engineers, applications engineers, and ultrasound experts. And we make benchtop imaging tools for life science researchers and drug developers. These are two of our flagship products. Tomek's going to talk a little bit about it over the next few minutes. But we are a hardware and software company, and we make tools that generates data that is critical to advancing therapeutics through their preclinical development. So that's where we operate. And we've been at this for about 10 years. This is me and my former PhD advisor, Dr. Dayton. The company was started out of UNC Chapel Hill in North Carolina. And initially the company was focused on oncology tools, though about 5 years ago, we identified this really neat opportunity for liver disease researchers. And that's what we're talking about today. And Tomek really led the charge there. So he's the CTO of the company and has been PI on multiple NIH grants related to this liver disease application. So we really brought it from concept through making it a reality, making it a product, and then partnering with our customers actually to use the product to generate data for their studies. And that's some of what he's going to present today. But yeah, no better person on the planet to talk to you about this than Dr. Tomek Czernuszewicz. He has over a decade of elastography expertise, and yeah, so give him tough questions. He can take it. Again, my name is Ryan. I'll be sitting on the Q&A box. If anybody has any questions, I might be able to answer them as we go. But Tomek, go ahead and take the mic, and we'll talk to you soon.
Yeah, thanks a lot, Ryan. Really appreciate the introduction there, and thanks everyone for listening. This has really been kind of a pet project of mine since I joined the company, basically bringing elastography to the preclinical world. But this slide sort of captures the higher level agenda that we'll talk about on the webinar today. I'll start by just sort of giving an overview of the available research tools that we can use for studying Nash and liver disease in general. And then we'll kind of talk about where SonoVol fits and how our technology is different, and where we think we can do a better job than what is being done currently. And then the third section here will probably be the most interesting. I'll try to summarize the experimental liver studies that we've done so far, and we'll round out with a Q&A session. So the primary focus of this talk will be on Nash, although I do want to make the caveat here that these tools do go beyond Nash. Nash is particularly interesting, though, because it sort of has this multi-stage pathologic process where initially we have a normal liver, but then due to lifestyle decisions or genetics, the liver begins to sequester fatty acids and enters this steatotic state. Now, what's challenging about Nash is that simple steatosis is not in and of itself bad. People can live with steatotic livers for many, many years, and only a small percentage of them actually sort of translate or transfer into this inflammatory state where the liver becomes steatohepatitis. And in this case, we have inflammation that starts to happen in the liver. Inflammatory cells migrate into the liver, hepatocytes start to die, and fibrosis or scar tissue is laid down. And if this process is allowed to go without any kind of intervention or treatment, eventually the liver is going to fail, or the patient is going to need a transplant. So in terms of the therapeutic approaches, we can actually try to intervene at any stage of this process, and all of these have really been tried to date. We can try to create anti-steatotic drugs. that prevent the sequestering, with the thought there that if we can prevent the fat from getting in, then we can curb this whole process. We can also try anti-inflammatories or even anti-fibrotics. The liver is pretty well suited for regeneration. So if we can prevent the scarring and the process that happens, perhaps the liver can recover. But what has been a challenge in the Nash pharmaceutical community, there have been a number of high-profile failures of drugs that have made it into phase 3 that looked very promising through the initial stages, but for one reason or another have not really succeeded in getting through the regulatory approval process. So there's a lot that we sort of don't understand in the with this disease process and how best to tackle it. And so with the technology that SonoVol is developing, we think that we can really come in at an early stage and help pharmaceutical companies and drug developers, understand at the preclinical stage what's happening and make, I guess, better decisions on, what mechanisms and modes sort of make it to that next stage and essentially fail drugs faster. So what does the landscape look like here in terms of the current tools? It sort of breaks down into these 4 major buckets, or really 2. We have, how do you know what's happening in the liver? We have invasive tissue sampling, which is covered by histology or blood biomarkers. And then we have non-invasive techniques, which really rely on imaging. So in this case, the liver is best suited by imaging with MRI and ultrasound. The preclinical world, researchers that are studying small animals, they really live in this world. The imaging side of things has really made a huge impact clinically, but preclinically, there's not too much imaging that's happening, and we think that's a roadblock. We think that slows down the research pipeline, and so we've designed a new imaging system that we think can sort of fill that void that may be present in the preclinical imaging for liver disease. Histology, of course, is the gold standard. There's nothing better to understanding a disease than taking the organ out of the body, dicing it up and looking at it under a microscope. We can stain for any kind of marker that we want to stain for, look at molecular mechanisms. Of course, the big problem with histology is that it's terminal. So we no longer have any sort of longitudinal evaluation of how the liver is responding through time on a per individual basis. So we have to make an assumption that a cohort of mice is behaving the same way. There's some other challenges related to histology in terms of time requirements and cost. And then there can be sampling errors that can happen if you don't properly sample the whole liver. But really, not having a longitudinal measurement is a problem because it does not really truly show you the dynamics of what's happening. A corollary to the histological outcome that's been developed is to take biopsies. These are, of course, the primary standard of achieving a histological outcome for humans. For mice and small animals, this is not as well used, mostly because it's very challenging to do, and repeat sampling is not actually encouraged. So the mice don't handle the biopsies very well, and it sort of is a one-shot thing. So yes, you can get non-terminal histology from mice through a biopsy type mechanism, but it's not really giving you a longitudinal evaluation. So the primary longitudinal method that's been used preclinically is looking at biomarkers. So these are minimally invasive techniques. We're still taking blood, but we can do it over time with a frequency of 2 to 4 weeks and look at some of these serum markers that actually have really nice corollaries in the clinical world. So ALT, AST, we can measure these in mice and those show us similar things that they do in humans. Some of the drawbacks, though, of the blood biomarker approach is that these types of biomarkers are usually general. They're sort of non-specific. They're general biomarkers for liver dysfunction, and they don't have any spatial sensitivity. You're essentially looking at blood and The consequence of this is that they tend to have variable sensitivity and specificity if you're trying to do very precise staging of your animals. And they can also be influenced by various experimental factors and collection processing methods. So trying to understand what's happening inside of an organ through a biomarker is just a hard problem in general. And that's where imaging comes into play. So imaging allows you to do non-invasive assessment of the organ of interest, in this case, the liver. And we can evaluate something not only over time, but spatially as well. MRI is really the primary gold standard for non-invasive imaging of the liver. It can do full 3D acquisition. It has excellent soft tissue contrast. But as we all know, MRI is very, very expensive and also slow. And time is money. So if you have a slow imaging system where it takes 30 minutes to an hour to scan a single animal, and you have cohort sizes of 60 to 100 animals, it becomes prohibitive. And demand is very high. Everybody wants to use their MRI. So although MRI is very, very nice and a very excellent tool, it largely gets unused in this space for the cost reasons, but also the fact that MRI, when looking at fibrosis specifically, has really struggled. People have come up with a new technique that I'll talk more about, called elastography, MRE. The E stands for elastography. This looks at the stiffness of the liver. And this is something that has come about in the clinic as well, and is the gold standard for fibrosis staging non-invasively in the clinic. In preclinical world, though, doing MRE is actually pretty challenging. You have to insert a needle into the mouse's liver to achieve the waves phenomenon that you need to make these measurements. So again, not a very accessible technology, unfortunately. Ultrasound is an interesting alternative to MRI. It also has good soft tissue contrast, perhaps poorer image quality than an MRI, but better contrast measurements than something like X-ray, for example. It actually has a very high frame rate. So the potential for ultrasound to collect data really, really fast is really, that doesn't have a comparison. Ultrasound can easily capture hundreds of frames per second, which is not something you can do with other types of modalities. And the equipment costs are relatively low when comparing to $1 million MRI magnet. But the big drawback with ultrasound, and one of the challenges, particularly preclinically, is the handheld form factor. So what that means is that because it's handheld, we now have a limited 2D field of view, and we need an expert sonographer to place this probe in the right spot every single time. Otherwise, we incur inter-user variability. This is true clinically, but clinically they have very highly trained professionals that spend their whole lives and get certified to become sonographers. So that's kind of how they've gotten around that problem. But preclinically, it's not as defined of a job, and there's fewer of them. And so the people that tend to take these measurements will be postdocs, grad students, people that may not have as much experience with sort of the manual aspects. The other problem with a manual approach is that the workflow can be cumbersome. And so the speed advantages that we get from this high frame rate modality tend to get lost when you have to do a lot of manual positioning. You're going one mouse at a time. You have to use ultrasound gel and sort of position things, and it ends up almost equating to the same amount of scan time as an MRI. Elastography has also been implemented for the ultrasound world and has really taken over the clinic and had a lot of success, but the preclinical options are very limited at this point, and follow the same motif as a handheld form factor. So we sort of looked at this landscape and said, can we do this in a better way? Can we achieve the same accuracy and repeatability and sort of non-invasive longitudinal measurements as MRI, but can we do it with ultrasound? And so SonoVol, what we've developed, and I'll talk more about this, is really a robotic platform to capture ultrasonic data, both traditional ultrasound data, but also these more advanced elastography approaches. And we think that this is a nice solution for preclinical imaging specifically. So the rest of the talk here, I'll sort of talk about the key aspects of our approach. We'll start with the, how we're doing this robotic scanning. We'll spend a lot of time on shear wave imaging because this is something that's pretty new and I think will be very powerful. And we'll conclude looking at our microvascular imaging modes, as well as some potential for our technology to merge with optical imaging, like fluorescence imaging, for example. So I alluded to this on the previous slide, but conventional ultrasound follows this handheld motif. Preclinical ultrasound really followed the same motif. They took the handheld ultrasound transducer, they increased the frequencies so that you could see smaller and smaller organs, but the onus is still on the user to have expertise to A, know what they're looking at, and B, put the probe in the right spot. Our approach has been different. We said, let's try to make this robotic and more push button, more like a Xerox machine. You put your animal on the pad, or more like an MRI machine, where you put the animal inside the machine, you click a button, and you get a 3D image. And this is the tool that we've designed. We call it the Vega. It's a benchtop instrument about the size of a laser printer. And it's just like a Xerox machine, as I mentioned on the previous slide. So the idea here is you put an anesthetized animal on top of an imaging membrane. All the ultrasound hardware and transducers lives inside the box. And we can do multiple animals that can be staged and posed on the instrument at a time, making for a very rapid throughput. What's inside the box? So what's happening is that we essentially have a robotically actuated carriage that holds one or more transducers. And to date, we've integrated 2 different transducers that have sort of different application areas. We have a wobbler transducer that's higher frequency, that's nice for superficial cancers and kidney disease. And then we have a linear array that can look deeper into tissue and to evaluate cardiovascular pathologies, as well as liver disease, which is what we'll be talking about today. This carriage translates underneath the animal in a fluid-filled chamber to create an acoustic path for the ultrasound. And all we have to do is fill this chamber with a thin layer of water, and we're ready to go and cycle through animals. This video shows how the raster scanning approach is done. So what's happening is, as the robot is moving the transducer, we're collecting real-time frames from the ultrasound transducer and building up a stack of images. This stack of basically thousands of images then get reconstructed into a 3-dimensional volume. And from there, we can create images that look nothing like a traditional ultrasound image. So here, what you're looking at is an example of a coronal slice through the 3-dimensional data set. And as will hopefully be obvious, you can immediately recognize some of the anatomical features that are typically hard to see in a traditional 2D ultrasound image. So in this case, we can see the spine of the mouse. We can see the 2 kidneys. In this case, we had a tumor-bearing animal. So this is a tumor that's in the side. And the workflow here really happens at a rapid pace. So the idea here is we have a camera that's looking down at the scene, and in the software, we can monitor the position of the probe and define a region over which we want to scan. And so as soon as the first animal is done, the probe will move on to the second animal. And the first one could already be woken up from anesthesia and taken off of the pad if no more imaging modes want to be captured. So the initial application area for this tool was really in the oncology space. We have this high throughput volumetric measuring tool. We can measure tumor volume, and we've done a lot of work here validating the system, comparing it against MRI for volumetric measurements, and we've demonstrated nice correlation. Here you see some examples of sort of different imaging poses on the system. Here's the liver up here, this dark area. We can see the stomach. The tumor is here on this particular subcutaneous model that we were looking at. You can see the legs, and really pretty easy to interpret what's happening in these images and know what you're looking at when you have this wide field of view. And so for the rest of the talk, I'll focus on liver, but I do want to highlight here that this is a platform technology. So Really any soft tissue organ that ultrasound can image, we are striving to image it as well. So cancer, heart disease, kidney disease, pregnancy, all of those things can be imaged with our platform. Okay, so, oops, sorry. So, to summarize, the robotic bottom-up approach really gives us wide field views of the tissue, rapid acquisition of the data with minimal expertise. So you no longer need years of training or a PhD in ultrasound to collect highly repeatable and reproducible results. Okay, so switching gears here to shear wave elastography, I'll give a little bit of background for those that are not familiar with this technique, since it's not really utilized in the preclinical research world. Elastography is essentially a technique to evaluate stiffness of tissue. And looking at stiffness as a corollary for disease state has been utilized for millennia by physicians. So, and really it's known as palpation. Everybody that's gone to the doctor has had this done to them. Essentially what the doctor does is place their hands on the abdomen and they compress the tissue to see how hard or soft it is. And what they can tell from palpating the liver in this case, or any other organ that's accessible or superficial area that's accessible by their hand, is they're looking for enlargement of the organ of interest. And they're also looking for any kind of firmness or hardness changes that may be happening. The firmness and hardness changes really come from the underlying inflammation and fibrosis that could potentially be happening under the hood. The challenge, of course, here is that, okay, we could do this in mice. You could go through and squeeze every mouse, but it's a qualitative measurement, right? So elastography, or the imaging approach to it, is really trying to put a number behind that stiffness to make it more quantitative and sort of reproducible across any person or operator or model. And so what we sort of get with elastography here, this is showing a series of phantoms that go from less stiff to more stiff. And what we tend to do is color code each pixel in the image. It's no longer the anatomical grayscale picture that you're used to seeing, but rather a modulus value, which is the engineering unit of stiffness. So in this case, a low value of modulus indicates things that are soft, and the higher that the modulus goes, it indicates things that are getting stiffer. Okay, so that's what elastography is, which we're essentially trying to quantify stiffness and use that as to infer disease or pathologic state. So what's the shear wave business that we're talking about at the beginning of this? Well, so it turns out that it's not trivial to map the stiffness of a tissue with an imaging modality. The shear wave is one approach, and is probably the most dominant approach that's used today in the clinic because of the advantages that it has over physically compressing the tissue by hand. So a shear wave is really just a mechanical wave. It's not a sound wave. So it's different than the ultrasound wave that's creating the grayscale image. It's actually a mechanical wave that travels laterally to the direction of travel of the ultrasound wave. And so everybody's kind of experienced a shear wave. If you've ever, basically hit a bowl of Jell-O and you see it vibrating, those vibrations are shear waves. They're mechanical waves that are traveling through that solid media that travel at a certain rate. What's nice about shear waves is that the speed of the shear wave is directly linked to the stiffness or the modulus of the tissue. So in this case, what we're showing is we've excited a shear wave in the liver, and in green here, we're showing the wave travel through the liver at a certain rate. The slower the speed of the wave, the softer the tissue is, the faster the wave, the harder the tissue is. And so now, in order to quantify this and create, instead of looking at a movie, which is, not really showing you a velocity, it's just showing you the wave itself. What we do is, on a per pixel basis, we can estimate the arrival time of when the wave gets to each pixel. We know the delta t from the different arrival times, and we know the pixel dimensions, the delta x. The delta x over delta t gives us speed. So for every pixel, we can compute a speed of when the shear wave is getting there and how fast it's going. And then if we make some mathematical assumptions about the underlying material that the wave is traveling through, that it's isotropic, that it's perfectly elastic, that it's linear, with a very simple equation, we can convert speed into Young's modulus, which is again, the engineering unit of stiffness. The equation is given here. We essentially square the shear wave speed multiplied by the density, which in the case of liver is very, very close to pure water, and multiplied by 3. And so, as I mentioned, these systems have really dominated the clinic. There's a whole host of commercial technologies. If you do a PubMed search for either shear wave elastography, transient elastography, or I didn't do it here, but magnetic resonance elastography is the third one, you'll get over 5,000 PubMed hits. It's hugely impactful and utilized not only for liver disease, but a whole host of pathologies, cancer, kidney disease, et cetera. It's a really powerful technology. Unfortunately, it sort of skipped over the small animal world. And so one of our goals as SonoVol is sort of a reverse translation of this technology and bringing it back into the small animal world. A few of these commercial clinical players do have solutions for small animals. But again, what they've done is they've taken their handheld form factor, miniaturized it, and said, okay, do your best to collect these measurements reproducibly. All right, so that's the background on shear wave technology. Now I'm going to transition and let's talk about some data here. So I'll spend a lot of time talking about this very first validation or experimental cohort that we did in a choline deficient high-fat diet model. This was one of our first studies, and what we were trying to prove here is essentially that the technique is correlating with underlying histology. So we started out with a group of 40 mice with 8 time points and that we were imaging over, starting from 0 prior to starting diet all the way up to 15 weeks. And then at every time point, we were sacrificing 5 animals at a time for a histological gold standard. And this 3D movie kind of shows you the highly steatotic and inflamed liver. That was those very bright pixels that you're seeing as it's sweeping through. Again, the parameters that we measured here are both the brightness of the image that you saw as well as the stiffness. So if we look at some representative examples here, on the left, we have a control animal. A control liver is generally pretty dark on ultrasound. There's not much backscatter that's happening. And the volumetric measurement is on the order of 1,000 millimeter cube. After 1 week on CDA diet, which is a really short time, you start to see brightness changes happening on the B mode. So it may be hard to see on the slide here, but the liver has actually gotten slightly brighter on B mode and slightly bigger. By 2 months on this diet, it's night and day difference. The liver has lit up like a Christmas tree. It's very bright and it's gotten very big. And this is expected as the liver has become highly steatotic, highly inflamed, and is already starting to develop fibrosis at this point. If we look at some representative shear wave images, this is kind of what they look like. The color map here is overlaying the shear wave speed pixels in this case. And in the control time point, we see that most of the liver is sort of this dark blue pixel around 1 meter per second. At 1 week, there hasn't really been much change. You can see the brightness is brighter as shown in the previous slide, but the stiffness has not really changed that much. But by 2 months, we're already starting to get into these teal yellow colors and the liver is stiffening up over time. So today, the system works by taking individual 2-dimensional frames and sort of raster scans through different fiducial locations, but we will shortly be releasing a true 3D shear wave mode that operates just like we do with the B mode, where it sort of sweeps through and stitches the images together. Okay, so in this particular model, here are the results for both echogenicity and stiffness. And this is really interesting. So if we look just at the echogenicity, which has the best correlation with the steatotic development, what we see is there was this very dramatic and quick rise between 0 to 3 weeks, and then we hit a plateau. Histology demonstrated that by 3 weeks, we were essentially maximum S3 steatosis. So this is the time course over which it takes these livers to essentially max out when it comes to fat content. On the stiffness side, though, we had a much slower progression in the increase in the stiffness, and this is consistent with the slower onset of fibrosis. So at the early, actually, the trend ended up to be very linear, which was pretty nice. But at these early stages between 0 to 3 weeks, we had sort of a moderate increase in stiffness from about 5-6 kPa up to maybe 7-8. But then by the end of 15 weeks, now we have really dramatic change in the stiffness, almost up to 20 kPa, or 4-fold increase in the stiffness difference in these livers. If we look at the underlying pathology, which was scored, or histology, which is scored by a pathologist, we see very similar dynamics. So again, the steatosis maxed out by week 3 and then stayed maximal throughout the study, whereas fibrosis had a much slower and sort of linear progression here over time. For steatosis here in this particular study, we actually added a 4th category. Normally the scoring goes S0 to S3, but what we realized is there was a zonal patterning that was happening with this particular CDA model, which actually ended up having sort of a higher level on the imaging metric than was sort of reported. But if we didn't have this particular extra grade, essentially all mice would be S3 by 3 weeks here. If we correlate the grayscale images, the imaging metrics versus the histological metrics, we saw actually very nice correlation between the two. On the left here is our echogenicity versus steatosis, which R squared of almost 0.9. You can see the progression through the various score levels here. And then on the fibrosis side, slightly lower correlation, although still very good at 0.84, with the scale ranging from 0 to an ESHAC level of 4. So one thing we did not observe in this cohort was ESHAC 5 and 6, which are the two cirrhotic phenotypes, or I guess scoring criterias on the ESHAC score. But we did have a pretty nice distribution of animals that spanned the 0 to 4 range, sort of the pre-cirrhotic ranges. I went through and actually performed an ROC analysis to see, what is our sensitivity and specificity or classification ability of the technique, because ultimately what we'd want to do is use our technology on the front end of a study to sort and select or recruit mice into various arms, right? So we want to basically create a fibrosis selected treatment arm that we can then hit with our drug and essentially reduce any kind of intragroup heterogeneity. And the ROC analysis demonstrated actually very good area under the curves, exceeding 0.95 in basically all cases. The sensitivities and specificities were nearly all over 0.9, and that's on a per ESHAC basis. So if we are trying to discriminate ESHAC 1 from 3, we had basically perfect classification sensitivity. So that was very encouraging to suggest that we can use this technology to basically classify and stratify the different levels of both steatosis and the fibrotic scoring as they do in the clinic. Okay, so the next slides here, I'll quickly go over some further validation studies that we've done so far. Some of these are actually ongoing studies. These are, there's a whole host of animal models that are out there that span various types of liver disease. We've performed some analysis through collaborators on carbon tetrachloride, which is the very classic model of fibrosis. We've been running a Western diet study. So this is a obesity-driven type Nash model. And we've also looked at some genetic knockout mice here. So this is the ABCB4 knockout, which is a model of cholestasis here. We've been serially scanning these animals to see, how does the parameter and the imaging metrics change with the different underlying mouse models. So in this slide, the top row will be stiffness, the bottom row will be echogenicity, and then we'll kind of go through the different models here. But so carbon tetrachloride, develops very rapid onset of fibrosis as expected. And what we saw here is that indeed we saw a nice rise in stiffness as measured by our imaging technology. And no corresponding rise in the animals that were injected with simple corn oil. Interestingly, we also saw a rise in echogenicity. I was not expecting this, although it has been reported after I did a review on the literature that the brightness of the liver actually does go up when utilizing carbon tetrachloride. And I have some hypotheses here as to why that's happening. Primarily, what I've seen is that steatosis is the driving factor of the echogenicity, but in the case of the carbon TET model, I think that the difference in the way that the fibrosis is deposited could be driving some of this. But nonetheless, either parameter is showing really nice discrimination from the control animals, even as early as a couple of weeks. The Western diet model, I was actually a little disappointed when I saw this data at first, although I do think it highlights the importance or the power of the imaging modality. So here in the stiffness map, we're really not seeing too much difference. The model has been running for about 40 weeks now, so almost an entire year. These mice have been on the Western diet. We've seen a nice increase and sort of asymptotic rise in the brightness, suggesting that the livers are becoming highly steatotic, but the stiffness has not really increased, suggesting that probably most of them are facing simple steatosis as opposed to slipping into that inflammatory and fibrotic Nash state. So again, I was originally planning to run this model out to 48 weeks and then do a takedown and the study would be done. But looking at this data, I might actually want to run this out longer. We've been running these mice for so long that it's, we've invested a lot of time. So I think that, although the stiffness increase has not really been that exciting, it does highlight an important use case of this technology, because otherwise you would just sack the animals and perhaps not have the level of fibrosis that you wanted by the end. The genetic model has also been very interesting here. What we observed is that, so these are mice that are bred and have either the gene, the wild types in the dash line here or the knockout here in the top line. So even at baseline, two mice that are 6 weeks old between the genetic knockout and the wild type already have a significant difference in the stiffness of the liver. There's much more marginal difference in the echogenicity, that the genetic mice do seem to have slightly brighter livers, but there's no real trend over time. Whereas in the stiffness, there appears to be this increasing trend over time, which would be consistent with the fibrotic development from the cholestasis that is present in these animals. I dug a little deeper into these error bars. They were a little large for my taste, but what turns out that if I stratified the animals by male and female, actually saw pretty different dynamics between the two. So in this case, the male mice had softer livers, and the female mice had stiffer livers, suggesting that the female mice were actually more affected and potentially more diseased. This is counterintuitive to what we typically expect for Western diet models, where the male mice tend to be more susceptible to the Nash phenotype. This is Nash, whereas this is cholestasis. But after speaking with our collaborators, it appears that female mice, this particular strain, actually do exhibit a more advanced phenotype. So this is, we still have to validate this with histology, but it's encouraging that the trends seem to be matching, and again, highlights the importance of, the imaging, it can actually differentiate some of these dynamics that's happening in the different models. So to summarize the sway section here, I believe, and I think this is true in a lot of the ultrasound community, that sway is a very powerful modality and should and could be useful for preclinical liver disease research as well. And we'd also like to apply sway to things beyond the liver, kidney fibrosis, tumor stroma assessment. These are things that we're actively working on. It allows us to quantify tissue stiffness, which has a high correlation to fibrosis and just general disease state of the liver. and will likely or can likely be used as an endpoint measurement for a response to therapy type study. And when used in conjunction with B mode, we can evaluate these sort of interesting interplays of steatosis and fibrosis, via echogenicity and stiffness mapping, which would probably not be possible with other types of approaches. Okay, so the next section here is talking about our microvascular imaging approach. SonoVol was actually founded on this concept of can we utilize injectable contrast agents and create very detailed maps of the underlying microvasculature. So in ultrasound, contrast agents are lipid encapsulated gas microbubbles. And the unique technology that Ryan, our founder, created when he was in grad school was this approach of basically superharmonic imaging of these contrast agents. So the idea here is that we transmit with a low frequency and we receive at a much higher frequency, almost like a second harmonic generation imaging that you would do in microscopy. Only now we're not looking at just the second harmonic, but huge harmonics that are fourth, fifth, sixth, and beyond. And the reason this can work is because microbubbles, when they're excited at low frequencies, have a very, very broadband and superharmonic response, and whereas tissue does not. So if we filter out the lower frequency excitation pulses that we're sending, what we're left with is the signal that just originates from the bubbles. And the microbubbles are limited to where they can go. They can only stay in the vasculature. And therefore, we can create these maps that essentially show us what is happening with the microvasculature of the tissue, which the B mode cannot show us. Although to date we haven't done a fully fleshed out longitudinal study, as I've done with the echogenicity B mode and sway imaging modes, we have taken some pilot data in livers and are observing really nice trends here that I think will translate when we do the longitudinal methods. We can see here the liver vasculature can be mapped out, and the architecture of the liver can be seen with this approach. We've also utilized this for evaluating liver cancer. So it turns out that the liver is so highly perfused that anytime a tumor, these are metastatic or tumor liver metastases from a metastatic model, it actually creates a void in the perfusion map here. So these METs can be quantified by looking at negative contrast if HCC is something that is of interest. We've done some validation studies. As I said at the beginning of the talk, oncology was one of our primary focuses. We've done some validation studies demonstrating that these different vascular parameters can be used to evaluate response to treatment. In this case, we were looking at treating with anti-angiogenic therapy and demonstrated that we can sort of visualize the treatment working and destroying that vasculature in the tumor. So microvascular imaging is sort of another avenue and another readout. Perfusion has been demonstrated in the literature to decrease as the liver becomes more fibrotic and diseased and damaged. And we also have portal hypertension, which is, can influence the vascular perfusion characteristics of the liver. So we think that this can be basically a third readout here, in addition to the two that I presented before. And lastly here, we have a nice opportunity with our technology to merge with optical imaging. So this is a new system that we've released recently, where essentially we have ultrasound coming from below the animal, and above the animal, we can do traditional fluorescence or bioluminescence imaging. So this is one of the advantages of doing bottom-up imaging, is we freed up the space above the animal to combine and do the top-down optical imaging, as you would see in a bioluminescence system like the IVUS. And so, the main primary advantage here is that the optical side of the coin really gives us excellent molecular imaging capabilities. We can generate fluorescent probes. We can do genetic knock-in models that have luciferase or luciferin expressing knock-ins. And with that, we can create these multimodal plots where we're looking at both the molecular signal, which is highly sensitive, but perhaps it doesn't have very good spatial resolution. And we combine that with ultrasound, which has somewhat less of sensitivity to individual tumor cells, for example, or individual macrophages if you're looking at inflammation, but has good anatomical description. So we can do 3D volumetric measurements with ultrasound. This was a study done in a deep pancreatic tumor with the combined method, showing that the ultrasound shows a sort of a smooth growth curve here, whereas the bioluminescence is quite noisy. So in this case, this could be done for liver cancer as well. And the main, this is to kind of conclude here, this is a final work that we presented recently at the WIMIC conference this past month, is utilizing both optical and ultrasound to look at the big three of Nash. So inflammation, steatosis, and fibrosis. I did not talk much about inflammation when I was talking about the shear wave and the echogenicity side of the coin. And that's mainly because those two technologies are really, that we don't have the resolution to see individual immune cells trafficking into the liver with those technologies. But with optical, we do. And so this was a collaboration that we did with the scientific group at PerkinElmer, the developers of the IVIS, where we essentially imaged mice, high-fat diet and carbon tetrachloride, so combination high-fat diet carbon tetrachloride mice with fluorescent markers for inflammation, as well as sway and echogenicity. And indeed, we saw rises in all three parameters, which is expected as these mice are going into this Nash phenotype. So we're excited to explore the multimodal aspects and essentially offer a quad modal, if you will, type system, where you have backscatter, stiffness, perfusion, and inflammation that can be mapped with these optical techniques. So yeah, the takeaway here on the optical side is we can really use optical to quantify the molecular inflammation or molecular information, which is specifically useful for looking at inflammatory states of the liver. So just some concluding thoughts here. We're kind of getting to the end of our time. What we're trying to do here with our imaging technology is not replace histology. Histology will not go away in the preclinical world. It is the best method for seeing what's happening at the cellular level. But we think that our technology really can complement histological methods, specifically when it comes to timing endpoints and sorting and recruiting animals for various treatment arms in order to reduce error bars. There are many, I alluded to this already, but there's many different animal models. Some are more expensive than others. Nash is a particularly expensive model to develop, especially if you utilize some of these more human translatable models like the Western diet model, where we're not messing too much with the genes or knocking out amino acids from the diet, which is not really super relevant for the human condition. But it's very slow. It takes a long time for these models to develop. More time means that the models are more expensive, and you don't want to be euthanizing your animals at a point at which it's the incorrect time to euthanize them or start your treatment. And then the imaging basically allows us to homogenize our treatment arms and basically do better science in the sense of being able to identify therapeutic outcomes with more power. So with that, I'll conclude, and I'll pass the microphone back to Ryan. Thank you so much for your attention, and happy to take any questions through the chat box.
Yeah, really great job, Tomek. Thank you for that. There's a handful of questions here, and we can probably get through them before the hour is up. So the first one has to do with, I guess, just like imaging parameters in general. It's two questions. Can you do it in 3D, and what is the resolution? So I suppose, yeah. Sway imaging or B-mode imaging, describe sort of the workflow there, and how small of a region can you get measurements from, I suppose.
Yeah, so the right, so I alluded to this when I was talking about the representative images. So right now, the B-mode images are happening fully in 3D, and they're very rapid as the robot is scanning through the volume of the tissue. The shear wave acquisition, the processing time and the data capture takes a little bit longer. And so the current method is to sort of sample the liver. We essentially use the robot to move to various places in the liver, and we take maybe, instead of taking thousands of frames like we do for B-mode, we take maybe 10 frames across the volume of the liver. And so we are getting a three-dimensional measurement, but in the current state that we have commercially released, it's a little more of a undersampled imaging scenario. But that being said, we're really actively working on bringing 3D sway to the market. In fact, our next software release will include 3D sway. And so, yes, the answer is that we will be able to do 3D whole liver sway is the goal with this next software release. In terms of the resolution aspect, yeah, so the B-mode resolution is going to be better than the sway resolution. The B-mode resolution, we're operating our transducers on the order of 24 megahertz. So we're probably talking about 100 micron resolution, something on that order. With the sway resolution, though, is probably closer to about 800 microns to about a millimeter resolution. So very, very small features will be sort of obscured in the sway image, but that's sort of less important for a diffuse, homogeneous disease like the liver, like Nash or liver fibrosis. But that being said, the different liver lobes can definitely be imaged at this scale.
Great, thanks. Question regarding throughput here. How long does this take per mouse? So I suppose like a workup or a scan of an individual mouse looking at steatosis or fibrosis. How long does that take?
Yeah, so the scan time is highly driven by the number of modes, number of parameters that you want to capture, and how large the mouse is really. So if you have a standard size wild type mouse with and all you want to take is a three-dimensional B mode, that data can be captured less than a minute per mouse. If you want to do both 3D B mode and sway, the sway adds another 2 to 3 minutes of acquisition time per animal. And then if you want to add on the perfusion imaging as well, that adds on probably another 5 minutes. You have to do an injection. So there's, the full workup is probably on the order of 5 to 10 minutes per animal if you want all three parameters. But, we're constantly working on ways to speed that up. And so part of this new software release where the sway mode will be put into sort of true 3D, it will hopefully speed up that process as well.
Okay, this is a question related to, I guess, clinical translation. Are you measuring this the same way as the clinic?
Yeah, that's right. So really the algorithms and the approaches that we've taken for, I guess this is specifically the shear wave mode is the most relevant here, since that's the one that's used heavily in the clinic. Those modes are very much using the same approaches. So we are in the same way that the ultrasound transducers in the clinic are generating the shear waves, we're doing that in the same way. And the tracking approaches where we're looking at the peak arrival times and some of the filtering approaches, basically all of that has been done consistently with what they're doing in the clinic. So our hope eventually is, to have a true mouse to man sort of system here where, or rather validation here, where, we can basically provide the measurement. And so that, we can use our technology as a platform for developing new clinical imaging technologies and hopefully improve the shear wave imaging that's happening in the clinic. That's the goal.
Great. And this is not a question, this is a comment. Nice talk. So
thank you.
I'd like to echo that as well. Thanks for your time here, Tomek, and everybody that tuned in. Thank you. And we'll put this on demand so you can share it with your colleagues if that's helpful. All right, we'll send that link around later to the registrants. All right, thanks so much. Why don't you wind it down, Tomek?
Thank you very much, everyone.